Okay. Let's wait a bit. It's not just noon now. So yeah, Jason, I'll start off with some framing about LDSC, and then you guys can take it from there.
Okay. And I am prepared to kind of start from the beginning too, so you don't need to worry too much, but yeah, that would be great.
I'm just giving a really brief introduction and where you can see the results in the portal.
Great. There we go. Yes, I finally saw Patrick. I'd be in trouble if Patrick weren't on.
Hi, everybody. Thanks for joining. We'll just wait a couple more minutes to let more people join.
All right, how much time do we usually balance towards like presentation versus feedback / questions?
It's really pretty variable. Sometimes there's more discussion and sometimes there's less discussion. So it's just really there's no format. So we do have this as a meeting format rather than a webinar format, so people can show themselves and speak when they have questions. I know, well, I like it.
I saw that. Did you forget to delete the eye? No,
I'm not telling.
Oh, I think that was her iPhone.
It is, but I forgot to delete the eye, which makes it even funnier. All right, I guess we can get started. And I'll start us off. So welcome, everyone. This is a focus group, so we hope that you will all ask lots of questions and be very interactive. Jason and Patrick are going to talk to us about implementing LD score regression in the knowledge portals. I do want to mention that we're recording this. Sometimes we don't record the focus group sessions because we really want your unvarnished feedback, and we still want your unvarnished feedback. But in this case, we're recording it because there is going to kind of be some instruction, and I've heard from people that they really want to hear this, but they can't be at this session. So we'll record it and post it. So I just want to start out with a really brief sort of perspective on LD score regression. So this is a method that was developed by Hilary Finucane and Brendan Bullock-Sullivan here at the Broad. And we use two variations of this method in the portal. So we use stratified LD score regression, and we use that to calculate global enrichment of genetic associations within genomic regions that are annotated as tissue-specific regulatory elements like promoters and enhancers. The idea behind this is that if genetic associations for a specific disease are enriched in annotations of a certain type in a certain tissue, that can give you some clues about which tissues and which regulatory elements may be most relevant for the disease that you're interested in. We previously used the Gregor method for this calculation, this global enrichment, but LDSC is more robust to confounders, and it also gives us more accurate and specific annotations. So let me share my screen and show you where in the portal you can see these annotations, these global enrichments. So one place you can see them is on the variant sifter. And I've shown this interface in this group before. You can find it under the KP Labs menu of the tools. And basically, when you enter a region of interest and a phenotype of interest, you will see a plot like this for global enrichment. So this shows you now the stratified LD score regression values for all of these different types of regulatory annotations in these different tissues. So the plot shows you which of them are most enriched. So for example, here we're looking at type 1 diabetes, and accessible chromatin in thymus tissue is the most enriched in genetic associations for type 1 diabetes.
And so another place that you can see these global enrichment values is on the phenotype page. So here we've got a phenotype page for coronary artery disease. And if we scroll all the way to the bottom, we will see the globally enriched annotations. So this is the same data that was plotted in this case. It's in a table, and you can sort and filter this table. So we see, for example, here that for coronary artery disease, enhancers in blood vessels are the most significantly enriched for coronary artery disease genetic associations. Finally, the second way that we use LD score regression is cross-trait LD score regression. And this takes pairs of phenotypes and compares their overall genetic associations. And you can also see this in the portal on the phenotype page. If you scroll up just a little bit, we have a table of these genetic correlations. So these values represent the phenotypes that are correlated most highly with the genetic architecture of coronary artery disease. Finally, you can also use these genetic correlations in the association of function knowledge portal. So here, this is our portal that shows all the phenotypes that we have. And you can narrow the focus of what you're looking at in this portal by picking a disease or a physiological system. But here, let's just pick coronary artery disease. And now, okay, we'll set the focus. Now we have, I'm sorry, I have to go to a region page. Now we're only looking at coronary artery disease phenotypes, but here we have now the option to set the focus by phenotype correlation. And when we do that, we can start with a phenotype. And then we'll see all the other phenotypes that are genetically correlated with that. And we can pick and choose which ones we want to look at. And then after we set the focus like this, we can see just those phenotypes in this portal. So that is all I have to say about this. That's my introduction. And please, oh, I see we have a question.
Yeah, I was going to say, Maria, we have a question.
Thanks, Mackenzie. Okay.
Yeah, so my question is like, so when you're doing the LD score regression, and like, are you able to tell whether there is sample overlap?
So I'm going to defer this question to Jason and Patrick because they are the experts here.
Yeah, so you're talking about for the genetic correlations, whether you
Yeah, so if you have two traits like she showed, where you have significant overlap, is it possible that maybe the same individuals were used for both traits so that you can have that kind of overlap, which is not real?
Good point. The answer is yes. You almost certainly do have that overlap. It doesn't detect the amount of overlap, but you almost certainly do. It's actually a good question. I'm 99% sure that sample overlap is okay and that this method is robust to that, although I should really just confirm that. And that's because even if you have the same individuals, the phenotypes are still somewhat different. And so we're looking at genetic correlations, and it's not necessarily like just because
I see.
But it's
so
actually,
it doesn't matter if there is sample overlap, it's still okay.
I'm almost positive about that. But to be honest, genetic correlations are a very complicated concept, something that even though I broadly understand, there's a lot of nuances to it. So I believe so, yes. Okay, so let me, so I'll take the lead from Maria here, and I'll go a little bit deeper into this concept of what we're trying to do here. Patrick and I kind of assumed we'd have a mix of people who are very unfamiliar with LD score regression and use the portal to look up genes, ranging to people who are like really familiar with the method. So please, we have everything kind of prepared. I'm going to start very high level. Patrick, I think he's going to trim his slides down to like 20 minutes or so, but we have information all the way into the guts of this method and all the nuances and how we're optimizing it. So we're happy to go in whatever direction that people want us to. Let me see if there's any questions here. Yeah, that's my understanding. Yeah. Yes. Okay. So let's go back to this page that Maria showed here. This is the variance sifter. And why do these enrichments matter at all? So as you all are probably aware, a big focal point that we're moving the portal towards in recent years is attempting to come up with new utilities to identify causal variants of the GWAS locus. So previously we focused a lot on if you have a gene, what is it nearby a association, or what are the associated regions for my trait? And we're now trying to move into within a region, what is the causal variant? And the major focal point in the field for doing this is collecting all of these annotations of like open chromatin and transcription factor binding sites and enhancers and various different cell types. And this review, I think, just came out yesterday from Kyle Galton, who curates all the annotations for our portal. And this is Nature Reviews Genetics, and they have this gigantic table of all of these various different, this is at the single cell level, but it also works at bulk level. I asked Kyle, we don't have all of these annotations in the portal, but we are in the process of putting them in. So, I think as you've probably seen before, you can on this page, look at all these annotations we have in the portal, of which there's, I think, about 4,000 of them. And you can see, like, if you highlight a variant, which annotations it overlaps. And presumably, if it overlaps in the right annotation, quote unquote, in the right cell type, that increases evidence that that variant is the causal variant. But the issue is with 4,000 annotations, like, how do you know which one to pick, right? You can see it's like a huge mess here. And if you've ever used the UCSC's genome browser, it's the same thing. We actually have too many, we simultaneously don't have enough annotations, and that we're not looking at all the right cell types in context, but we also have so many right now that, for coronary artery disease, which one is the right tissue to look at, right? And this is just very high level bulk tissue that, it gets even more fine grained at the level of like biosample. So we have this idea, it's not our idea, but I think something that people came up with, which is, well, a good place to start is an annotation that associations for this trait appear to occur more frequently than you might expect, right? So if the annotation is relevant to disease, you'd expect to see more associations in it, right, than by chance. And as Maria mentioned, the old method we had was called Gregor. This was getting kind of 8 years old. And the way Gregor worked was it just looked at the genome-wide significant variants and counted what fraction of those were within the annotation and asked whether that was more or less than expected by chance. And they had to do the stuff with LD and handle all that, but it essentially was looking at the most significant associations, whether they're enriched in the annotation. And this worked okay, but the issues with it were twofold. One is it only looked at the significant associations. So if you didn't have any significant associations, it didn't work at all. And even more generally, obviously, there are many other associations below genome-wide significance you might use. But more to the point, annotations are very correlated with each other. So an open chromatin region in adipose tissue is highly correlated with open chromatin in pancreas. And so almost every annotation was highly enriched, right? So if you ever used this feature before, you'd have like 10 to the minus 20 p-values for almost every annotation. So didn't do a very good job of like distinguishing which were the truly enriched annotations. So stratified LD score regression was this improvement to this method that looked at not just the genome-wide significant, looked at every variant in the genome, and Patrick will go in, I think, a little bit detail how that's calculated. And also, more importantly, it allows you to control for confounders. So you can calculate the enrichment of an annotation once you control for other annotations, okay? And that's called the baseline model that this group put together, which are like 100 or so annotations that are kind of consistent across all these different cell types, that what you're really looking for is, do I see enrichment in my cell type or annotation beyond what I expect from the basic background, okay? And so what we've basically done is Patrick has implemented a method that we now, for all 500 or so of our GWAS, we run across all currently 200 high-level annotations, so all the ones you see here, accessible chromatin enhancer, promoter, and binding sites across all these different high-level tissues. But we're doing a ton of work in going down even further. So you would get this not only at the level of, it's not in the portal yet, but you'd have enrichments at the level of biosample and even at the data set level. And so that's something we can talk about, and we'd love to get feedback as to how useful that would be and how far down we should take that, that process. And I think the only other thing I would mention, just because we'd love to get feedback on this too, is that these methods actually generated a lot of new results that I'm unaware if anyone's ever looked at before. Okay, so, we have, we're going to have for 4,000 annotations, enrichments across 500 traits. You're going to have genetic correlations across all pairs of traits that have, are newer than the previous analyses that were done when these methods were published. And what's also really interesting is that we have them at an ancestry level. So we have LD score regression enrichments, not only for Europeans, but if you have a GWAS that was in African Americans, we have enrichments that are specific to the African American GWAS. And I'm unaware of anyone who's ever compared, like, do you find the same things with African American enrichments as you do for European? And so we're interested kind of in feedback on whether there's any questions we could look at or how to validate this, or whether people are interested in this sort of thing. It's a lot of data and we'd like to start to make sense of it. And, I think Patrick will touch on this a little bit, but it was actually extremely complicated to produce these stratified LD score regression estimates for non-European ancestries because the input files were not available and Patrick had to track all of these down. So I'm going to turn it over to Patrick to talk a little bit about how we implemented this so you can get some more details on the method and our plans for it. But before that, I'll look at the chat and if anyone, this would be the time to ask any like high level questions, I think, about like the goal or in the intent and things like that. All right, genetic correlation. All right, I was wondering why in some they use the intercept of LD score to show there was no sample overlap, if I'm correct. I'm aware of the intercept of LD score. So you might be referring, David, to the original version of LD score regression, which is run on a single trait and there's a single value. And you basically, the way it works, I think, Patrick, I don't know if you'll discuss this, but you're regressing the observed like association strength on the number of SNPs that a variant is an LD with, and you expect some linear relationship with the intercept at, I think, 1. And because a SNP with no LD partners just has that reference value of 1. And if that intercept is greater than 1, it indicates that there's some artifacts that are like causing the associations to be systematically inflated. And I'm unaware if that's overlap. It's usually like population stratification or things that haven't been controlled for. And that was the original goal of this method, to distinguish that intercept term from the slope. Slope is the heritability. And formally, the enrichments are the degree to which heritability is overrepresented in annotation relative to your expectation. And then the intercept captures like residual confounding and things like that. I don't know actually what the intercept means for genetic correlation. I probably should, though. Okay, and I think that's all the questions. Do we have anybody want to ask a question high level before we get into the details, or post anything to the chat and we can also answer that? All right, I will.
Sorry, Jason, a really quick question. How do you guys deal with traits that have either weak heritability, either due to sample size or they're just not very heritable?
Very good question. Patrick, do we actually have a cutoff? So obviously the obvious thing would be you first run the baseline or the basic LD score and verify you have a significant correlation, and then you just don't proceed if that doesn't exist. But I don't think, Patrick, we have that cutoff, right?
No, the genetic correlations, the way the thing is built, it'll every so often complain about that it's detected something that doesn't really make any sense, and it'll spit out like not a numbers, and then those don't end up anywhere. Like those just don't end up as outputs for the system. So some of them won't have, like, they won't be correlated just because it didn't get a value for that pair because it was too weak. But the I don't think the LD score regression, every so often it will complain or give warnings. I don't know. I don't think it necessarily doesn't output anything. I think it just, you end up with a really low p-value or high p-value.
High p-value. Sorry, that was our experience with like the diabetic complications. I mean, they're already low heritability, and when you look at the cross heritability, you don't get very reliable estimates. The other question, I don't know if you guys have tried it, is I think the reference, the genomic, this is kind of getting into the weeds, was I think HapMap SNPs.
Yep.
Have you guys tried extending it to 1,000 genomes or had success?
So there's two. So so try to keep this in the weeds because I want to get into the weeds but for high level too. The way LD score regression has 2 steps. You first calculate the number of SNPs that are in LD with each SNP. Okay, that's always used the 1,000 genomes reference panel, and that's supposed to be like, because that's the proxy for the number of causal SNPs in the genome. And that is, that was actually Patrick. We did update the 1,000 genomes version as part of this, but that's always been 1,000 genomes. Then separately, now you have those LD scores for every SNP. You regress the chi-squared statistics on the LD scores. That has always been done using HapMap2 SNPs because those are considered to be higher quality SNPs. You could run it on 1,000 genomes, but I think since you already have enough power with just the 2.5 million SNPs, or you're just trying to fit the slope, it probably has a very small impact if you were to go beyond that, because the major goal here is just to get like enough high quality SNPs that your regression is a high quality. But that could be something we could look into, whether like, I'm assuming they did HapMap2 at the time because it was much higher quality than 1,000.
HapMap3, but yeah.
HapMap3.
We actually tried updating it on our end with like no success, but
yeah,
we might have missed something. So, but thanks for the explanation.
So I appreciate looking at minority group. Yeah, Eva, did you want to elaborate on this? Or
I guess back when VertaHealth first launched themselves as curing people of type 2 diabetes by weight loss, they were looking at glucagon also, fasting glucagon to insulin ratios. And it might be helpful under this phenotype, but the problem is I don't find that much research using glucagon values in. I asked several endocrinologists and they said glucagon moves up and down. Well, yes, but what I guess is when someone's fasting, a snapshot would be helpful.
got it.
So just an idea. When I looked at this phenotype, I thought of it, but whether you can get the data is a big question. Yeah, thank you.
Good point. All right, and I see Stephanie, you have a question.
Yeah, you were just saying that you get those correlations and then you regress them on the chi-square. Which chi-square for association is that? I just want to make sure I'm following the train.
You have a GWAS, right? So
Yeah.
like GWAS for fasting insulin adjusted BMI. Every SNP gets a p-value, right,
Yeah.
as part of it. And then you convert those p-values into chi-squared statistics.
Yeah.
So it's the
Okay.
association chi-square.
Okay, thank you. That's what I thought, but I just want to be sure. Okay, thank you.
It's very actually non-intuitive LD score regression, but the way you can think about it is that if you have two causal SNPs and then they're in LD with each other, everybody who carries one SNP will carry the other. So the observed effect will actually be double, right? Because if this SNP increases by one, this SNP increases by one, they're in perfect LD. Everyone who carries one will have the other. And so now they'll be increased by two. And so that's the intuition that the more SNPs that are in LD with each other that are causal, the higher the observed chi-squared will be. And that's the whole idea behind this regression. So more LD, you should see a higher chi-squared, and that is why that slope is heritability. There is like an assumption that every SNP in the genome is causal for this too, which is obviously not true, but like it at least works in practice. So you're always regressing the chi-squared from the GWAS, but the LD scores upstream are done from the annotations and have nothing to do with the GWAS at all.
Thank you.
Okay. Yeah. So, and actually, so one final point I'll make, because I wanted to say that, is that we're seeing a lot of questions about like the results here. This is absolutely something that like these results were produced a month or two ago. We want to have help in vetting these and figuring out how to do that. And so, yes, the T1D SNPs are predominantly lymphoid. What we hear suggests thymus. Like there's lots of reasons for that could be. We're looking for people who have interest in broad collections of traits who might help us kind of vet whether what we're seeing is actually correct. From a cursory glance, it looks very correct, but we're also seeing some very interesting results in some traits. Like my favorite example I've been doing is that oily fish consumption has like a very high correlation with bipolar disorder. It's like the third strongest correlation for it. And we're trying to figure out whether that's a real thing or what the causal direction is. And so those are things that if people are interested in helping us understand the biology here, we have some thoughts. And also one other thing just to put out there, which is that there are many other versions of LD score regression. This has been like an extremely fertile research area for the people who developed this, which was coming out of Alkes Price's group. So there's methods that do like calculate the effective number of SNPs in the genome, and methods that calculate cell types based on gene expression. And there are methods that calculate like, what else did I have? Like continuous values and things like that. And so we're considering whether adding more of these things might also be a value. I heard a question. I saw John Paul, you raised your hand. I don't know if you were the one I heard. It looks like John Paul's on the phone. Should we, anyway, okay, should we just go into Patrick and then John Paul, why don't you put in the chat when you're done with the phone call? All right, Patrick, I'll turn it over to you.
All right. Assuming everyone can see the slideshow. Still? All right, cool. Yeah. I'll be mainly talking about all the stuff, basically how we implemented LD score, all the different kind of issues we had while doing it, all the challenges and stuff like that, and then go into a little bit more detail than what everyone else kind of talked about. So this is straight out of the Bullock-Sullivan paper, which is on the basically like the tutorial page and where all the LD score software that we used came from. So basically LD score quantifies the contribution of polygenetic signal, and effectively by examining the relationship between test statistics and linkage disequilibrium. And just the only important part really is that it's the test statistics versus linkage disequilibrium, which is what's being kind of regressed in this case. And so, yeah, this should be z-squared. No, and it just occurred to me when Jason was talking. But basically what we have is, as far as test statistics, is our GWAS summary statistics, which are these kind of z-scores, the betas. And then it's basically just a simple linear regression versus genetic annotation linkage disequilibrium, these LD annot scores. And yeah, the nice thing about this LD score kind of method is that it operates on summary statistics. So it's not on the actual like genotype. It's on the summary statistics that you have in the GWAS. So it can be faster in that regard. And then as far as, and people touched on this, as far as its relationship to Gregor, there's this partition heritability or stratified LD score regression, which basically is trying to remove these confounding factors via a baseline model. And yeah, as for the results in the portal, there was basically three parts to it. We have our bottom line ancestry specific results, which are where the summary statistics come from. And then we also have the annotations, these genetic annotations, they come from CMDGA, and we have to transform those into our LD scores. And then there's the baseline model, which for a couple of the ancestries, we could have used the available baseline model. But in the end, and I'll touch a good chunk of it, the discussion will be about us creating our own baseline models. And part of that was because we have all these ancestry specific results for these ancestries where the baseline model basically didn't exist. Yeah, and just to put in a little bit more of a visual format, basically you have these GWAS, and there's basically 5 different methods that exist in the LD score that we used. 2 of them are pretty simple. So there's the Munge sumstats, and all that really does is transform the GWAS files into into a format where this Z variable is in a format that the rest of the software kind of knows how to deal with it. Similarly, there's this make annot, and that all that really does is transform the genetic annotations, which are in the form of basically ranges of the genome, so a chromosome and a start end pair. And that'll transform it into, again, a format that's just like a bed file that it knows how to deal with. And there's these 2 methods, the L2 and the H2 method in LD score. The L2 method is it's calculating LD score for the annotations from the bed file. And then the H2 is a multivariate weighted linear regression. And yeah, the baseline model, which is all of the stuff on the left there, LD1 to LDI, it's just trying to remove these confounding factors. So it runs this multivariate linear regression, and then you get the enrichment value just from the regression from the LD annot part at the end there. Yeah, just to go into each piece a little bit. Yeah, people maybe are familiar with, we use our bottom line method, which produces our ancestry specific meta-analyses. And so in the portal, it deals with a bunch of different studies. So you have a ton of, for type 2 diabetes, for example, you might have a ton of studies that have to do with EU ancestry or African ancestry or mixed ancestry. And so for each one, for each of the ancestries, it does a meta-analysis in which what it's trying to do is remove some of the correlation based on overlapping subjects, basically between the studies. And then for the mixed, it just picks the largest mixed. So you end up with these 6 to 9, I guess it's like 1 to 9 different meta-analyses or largest data sets that then you have available to do summary statistics on. Yeah, and then you take, for example, your EU ancestry-specific meta-analysis, and that's what goes through this Munge SumStats method that produces the first part of the regression. As far as CMDGA annotations, yeah, this was touched on a little bit, but basically there's a ton of information here. There's accessible chromatin binding sites. You have the tissues like pancreas, lung, but then things that aren't currently on the portal are things like biosample. So inside pancreas, for example, you can deal with islets or A cells or G cells, et cetera. There's 16 biosamples for pancreas, for example. And then you also have things like method source data set, which we don't currently use in LD score regression. And so, yeah, we currently only use annotation and tissue, which is 171, so about 200 annotations. But in the future, what we're looking at would be doing things like annotation tissue biosample, and there's about 1,800 of those, or going all the way down to annotation tissue biosample data set, and there's about 6,000 of those kind of individual partitions you can make on the 4,000 data sets that we have in the portal at the moment. And yeah, so the first part of the second step then is just reformatting that. So this is just an example of what the annotations look like. And as I said, they're just a chromosome with a start end is effectively what they are. And it has all the rest of the information that kind of dealing with what partition you actually are looking at. And the make anit just converts that into a bed file, which again, the rest of the software just kind of knows what the format is going to be then. And as far as the LD score, so like linkage disequilibria, on the left is this idea of you take 2 different SNPs in the genome and you can calculate a correlation between them, and that produces an LD value. As far as LD score, the idea is you take a SNP and then you are then comparing basically the overall correlation with the rest of the genome to produce an LD score. And what we're using here is it's one SNP versus not the rest of the genome, but it's the genome that's in the annotation that you're dealing with. And so that gives you an LD score for the annotation for a SNP. So what you're getting at is then a vector that is all of the LD scores for all of the SNPs that you're looking at with relation to the annotation. So that's what the L2 method in LD score does. And as far as the baseline, the why do we decide to generate our own baseline? Part of it was that we have different ancestries in the portal than was available. So what's available is European and East Asian, from what I know. But things like Hispanic or American and African and South Asian, there's no baseline available for that. So we were going to have to, if we wanted to get data for that, we would have to generate that ourselves anyways. We also had different G1000 references than they used in the original baseline model, which Jason touched on a little bit. Basically, we did that to update. We did it a while back to update a different thing, but also we just had a different, in the process, we generated our G1000 reference as well. And then it also allows us in the future to maybe add new variables or test some of the different variables in the system that that otherwise you kind of would have to just live with if you were going to use the baseline as it existed. And yeah, once you have the baseline, you now have everything you need to kind of run this regression. So all the Z's here, those come from the different traits, from the trait, the LD on the 1 through LDI comes from the baseline, as I said, LD anna comes from the annotation. So now you have everything to run this weighted linear regression and get the enrichment value. So yeah, just that this is like the overall system as it stands. Before I move on, does anyone, let me see if there's anything in the chat. Anyone have any questions? Oh, what is CMDGA? Jason, you wanna cover that? Oh, here we go. No, Jason already did. Never mind.
Yeah, he answered that.
Thanks. Oops. All right. Yeah,
John
so
Paul has a question, Patrick.
Yeah.
Okay.
Hey, Patrick. Hey,
Oh, sorry.
just a really quick question. So
Yeah.
you talked about the chromatin structure and accessibility. Are there other regulatory elements that you're also, I know that this talk is about the LD regression, but just a quick question, because these are other issues that affect regulatory processes. Is there anything else in there other than the chromatin structure and accessibility issues with the nucleosome? Do you have any other?
Yes.
Because
So we do have there's definitely histone marks that we have some annotations for, and there's computationally predicted chromatin state. And I'm sure there's others more. So I don't have a full catalog of it, but there's I can send you the link for seeing everything that there is on the portal. I do know, in general, the field has moved away from histone marks, I believe, and more towards open chromatin, but those still are available, like the ones that we have, and so there will be enrichments available for that too. And I think
because I guess in terms of the nutrients, I won't go too far sideways, guys, please. Is that, if we talk about methylation or acetylation, and we talk about, you gave the example of a fish, but if we talk about the epigenome and factors that certainly affect those processes, and I study diet, whether it's acetylated or methylated might make a big difference. So those were just questions that I had, but it's, again, to maybe a tangent. So I'll stop and we can maybe talk more about that.
Yeah, there are a whole host of annotations that are chromatin state. I do not know if those are actually, that's actually an interesting question. They're not available currently on that page that we showed, so we'll look into that. But we definitely have things that will have all the data for them, and it'll just be a question.
Because even the lysine that it attaches to, I mean, these are all pretty important, issues when you get down to the underlying biology. So thank you, guys. Thank you. Yeah.
Cool. Yeah. So as far as building the baseline, this probably was like, I don't know, 3-4 months of our process of getting this all onto the portal was spent just building these baselines. And the first part of it was just trying to recreate their European baseline from scratch to see, basically to track down all the input data so that you could do it for the other baselines like African, et cetera. And so, yeah, they do provide everything. They provide the baseline bed files, they provide their G1000 data set, and they provide the eventual baseline model that are generated from those. And so you can try to recreate it. And in the end, this is all available. Yeah, I'm gonna make the same joke I made before. This used to be free, you could just get it, and now it's not free. Now you have to pay for it. I think it's pretty cheap, but I'm only slightly afraid that I'm the reason why it's not free anymore. But they are available via a Google Cloud.
Is that actually is that the real directory name? Public requester pays? That's funny.
Yeah, I think so. Yeah. I can't remember. It costs a cent or a fraction of a cent per gigabyte or per something. It's not expensive, seemingly. Like it's not that big of a deal, but at the same time, like all of a sudden it costs money. I was a little bit surprised by that. That was a few months ago. But anyways, once you have that data, you can look, and there's basically 97 variables they have in the baseline model. That's all here. They're basically like different regions, so peaks or conserved regions or conserved primate, etc. But they also have these other ones, which are things like allele frequency bins. And yeah, these are just trying to the ones that are in purple. These are basically just direct translation. You take a bed file and you can just directly translate them into a binary variable. It's 1 or 0 depending on if it's in the annotation. The allele frequency bins they're splitting up the genome by allele frequency. And then in those 10, if they're in those each one, they get a 1 or 0 depending on which bin they're in. And that's the kind of trying to untangle whether allele frequency kind of affects the heritability. These ones in green, you have to use kind of an outside resource to calculate, or you have to calculate directly from the data itself. So like recombination rate, nucleotide diversity, et cetera. The ones in red, those are continuous variables. So they're not quite like the bed files, but they're very simple. You just translate numbers that they've calculated. So they're like EQTL data, and you just put those directly in there. So you can have continuous variables inside the regression. And then the yellow, these synonymous and non-synonymous, those are the only two we changed but kept in the model. And that was because they used ANOVAR to calculate, get those values, whereas we end up using VEP, and it seemed like VEP was a little bit better, or at least more updated. So ultimately, I kept them in, it's a slightly updated version of those variables, basically. And as you can kind of see there are 4 missing because there's only 93 in our model. And just to go through those quickly, one's, I call it GERP, it's probably called GERP, but those are 2 variables. One of was RS and one is NS. RS is the output, NS is the input, and the NS values are just totally unavailable seemingly online. And then when I kind of drilled down and tried to ask where this values came from, I end up learning that they updated the method, and the original NS values are now considered basically just like don't use them. So ultimately, I pulled out both of those until we can kind of figure out whether we want to use the updated method or how exactly we want to address this part of it. This ASMC one, I couldn't get the numbers to match exactly. And then there's also a question mark. I have to contact the original generators. It seemed like they say that it only comes from Dutch people, like the Dutch population. So I'm not exactly sure what that means as far as whether it can be used for non-European baselines, although they do use it in the East Asian baseline. So seemingly the original group was fine with it. And then the African LD. So this is, they're trying to remove the influence of kind of everyone's genome kind of goes back to Africa. And so they're trying to kind of remove the influence of that kind of ancient DNA from Europeans in particular, was like the original kind of motivation. But I found the methodology very confusing. I have to contact them about that. But also I really don't know whether you can even use it with the African baseline, because that doesn't really make any sense to remove African from the African baseline. But so yeah, those are the ones that are left out, and we just need to kind of follow up and see if we can't get those either added in or replace them or something. Yeah, ultimately, as far as our implementation is concerned, there's these 4 different parts. The bottom line, to some stats, that's really fast and pretty easy. The annotation to LD and the regression are both slow, but for 2 different reasons. So the LD is just slow, but it's operating on just like one time and you get all the data back. Regression is fast, but there's just, at that point, you're combining, it's a big matrix. It's all traits versus all annotations. So you end up with currently about 300,000 different calculations you have to do. And if you did it straight out, it would take about 2 minutes each, which already is kind of prohibitively expensive. And so I had to update the algorithm already to kind of get it so that we can get all those things through during this release. And then finally, because we do everything by ancestry, there's also a question of how exactly do we create then a overall value? And that is just a question of do we meta-analyze the results and how? And as yeah, these were just forked currently from Bullock. So we have our own forked kind of repository that has all these algorithms in it. And then we'll be looking to kind of, more specifically either refactor or figure out what to do with the method going forward. Yeah, and just a few notes, which I think could be of interest. So we applied the SumStats conversion to all of the ancestry-specific results, which isn't just those 5 ancestries. We have more ancestries in the portal. So there's like EU, EA, but then eventually you have also things like Sub-Saharan African or Greater Middle Eastern, for example. And then we also apply to the mixed ancestry. So overall, we have about 2,000 summary statistics files that we generate from the portal data. And as far as the regression, we then use the ancestry, for each ancestry specific meta-analysis, we use the closest G1000 baseline. So for Sub-Saharan African, you would naturally use African, but for Greater Middle Eastern, you end up using South Asian, which is how it's classified in G1000. And then for mixed, you end up using European, it just because most of the mixed data sets are mostly European data. And we don't currently have the information of exactly what components comprise a mixed data set in the portal. So in the future, as that maybe changes, we'll probably have to address exactly which baseline we want to use with each mixed data set. And then, yeah, as far as the meta-analysis, these are just the 4 things you can get out of the LD score regression. You can get heritability and enrichment, which we've talked about a little bit already. But you also can get the direct coefficients. These are basically the slopes from the linear regression. And then there's this concept, they just call it diff, which is they have this overlap matrix and they basically multiply the coefficients by this overlap matrix, and that produces a diff vector. And somewhat annoyingly, that's what they use to actually calculate the p-values for the enrichment. So it's based on diff, and they use a t-distribution using all of their jackknife values they get that they use to calculate the standard error for the enrichment. And so in order to do the meta-analysis, basically how we approached it was we combine all of the diff values for each of the ancestries using just inverted variance weighting. And then we calculate a new p-value based on that using the t-distribution and the same 200, whatever you call it, the free degrees of freedom. And then ultimately we either display the results from the largest mixed results or the meta-analysis. Well, I guess actually currently we don't do this. Currently we actually display everything. So the one that is called mixed is either the largest mixed result or it's the meta-analysis, whichever has the smallest standard error. And then we also display all of the individual ancestry results as well. Yeah, let's see. This is a good break point if anyone has questions. Anything I need to address, Jason?
No, I think I've been able to address them all.
All right. All right. Yeah, just to go here, this is effectively already heart failure. Oh wait, interesting. So yeah, as has already been shown, you mainly see the results are basically on two different places, but one of them is the phenotype. So here you can basically see the results for the but this is for heart failure for you know, muscle structure, heart, kidney, all these things kind of these all make sense as far as what you would imagine would be the tissues that would be affected by or associated with heart failure. Just to go to BMI. So these are a few of the ones that we use just to kind of test whether the method was operating as we expected. So you can see here, BMI, if you just look here, waist circumference, hip circumference, arm fat ratio, all this kind of like obviously makes sense. This is mostly BMI as well. Weight. As far as the enrichment, you have the central nervous system, pancreas, and eye. Those are basically the top three if you're looking at the mixed results. And the point I'm basically just making is if you go back and you look at the original paper, when they were developing the method, this is one of the ones they tested. And you can kind of see that central nervous system indeed is extremely associated with BMI. And so is pancreas. And then as far as eye, that could be an interesting one where I don't know whether that shows up as under one of these things like other, whether it wasn't part of their original test. I didn't look into it that deeply, but that could be something where that's interesting. If you look, I believe if you look around that the eye does show up as associated with BMI. So, and then a similar thing you can do with something like height, for example, where again, if you look at everything, if you look at the pathways, you can see all this stuff with like skeletal system, cartilage. For the associated traits, you got a bunch of BMI stuff, but weight, leg fat ratio, hip circumference, all this stuff kind of associated with height. Am I still associated with? No. And then down here, you can see adipose tissue, muscle structure, blood vessels, skin of body, connective tissue. If you go back again, the connective tissue, heart, these are all the things like skeletal muscle. These are all the things that kind of were associated with height. So it does seem like the method as we've run it on the portal seems to be reproducing the results that you would expect. And then also potentially, you know, with the eye thing, we might be seeing something that is potentially of interest to explore, would be the example. Yeah, just to briefly talk about kind of where we're thinking as far as the future is concerned. We're obviously going to be looking at validation. So one question is like, can we apply it to the meta-analysis in the first place? We're applying it to the bottom line meta-analyses, but we're going to be looking at applying it directly to data sets as well. But there's a question mark of like, is that going to be a problem applying to meta-analysis? It's something we have to kind of look into. How we do the meta-analysis, we might have to figure out whether that's the best way to do it, or is there a better way to kind of figure out what an overall number might be for the LD score regression to display. ancestry differences. It wouldn't be at all surprising, like the African LDD showed that, but you might actually want to have different baselines for different ancestries. It's just something that doesn't seem to have been done. I know that the European and the East Asian baseline that they provided are identical. They have exactly the same variables. So whether we want to have different baselines for different ancestries, I guess would be a question mark. And then finally, do we need to filter out non-GWAS? So currently these meta-analyses they have XGIP data, XSEEP data all in it. Whether that's valid is something that we'll probably have to look into as well. As far as the near future work, we're currently working on getting all those full annotations of things like biosamples in particular. In order to do that, we need to basically speed up the algorithm. So that's one thing that I'm currently very focused on, is getting the speed between once you have all the data, actually getting the enrichment value out. How fast exactly can we make that? And yeah, as I said, like these are the issues. Like LD is slow but one time, and regression is fast, but you have to do it a lot. Well, that's going to be exacerbated by this future work. We currently operate on about 200 annotations, but that's going to be at least 2,000 if we do biosamples. As far as the regression, it's fast, but there's a lot. But again, it's going to increase by an order of magnitude the instant we increase those annotations. So there's definitely work we have to do to make sure that it doesn't take 10 times as long to do all this stuff. And then as far as the longer term future, we obviously want to try to complete the model, get all the 97 variables that they had in the original baseline in there, and also potentially look into improving it. Then, as Jason touched on a little bit, get the cell specific and fourth moment, these other different variations. All the work we're putting into LD score, I think, should be easily kind of leveraged to make a bunch of these possible in the future. Yeah.
Great. Awesome, Patrick. And just one, to extend that a little bit, this came up in the chat, but there used to be this resource called LD Hub, where you could upload your GWAS, and I think it would run just the basic heritability, and also, I don't know if it would actually analyze cross-trait heritabilities, but we think we'll be able to get this fast enough that we'll be able to have a service where you can upload your own GWAS, and it will just automatically run LD score regression on it, and then also scan for enrichments across all, I don't know, 4,000 or however, I don't know how many we'll be able to do, but and we're hoping that we'll be able to provide that as a service for people, and you'll be able to get those like interactively. So that will be okay. So we've got about 8 minutes. So a question from Rainey. I don't know if that's directed just at me or unintentional, but any thoughts incorporating pleiotropy analysis into the portal? Yes, although it would be good if you have specifics. So what we're going to do next, I think, is Mendelian randomization as the next method. So we'll do not just the correlations, but we'll run the MR. And there's also another version by the people who wrote LD score regression we're exploring as well. So this like LCV model that's supposed to be a little bit like kind of more polygenic. But Rainey, if you have a specific method for pleiotropy that you think would be good, we'd love to hear that. We actually do have someone in the group who's working on that. So we'd be happy to consider that if you have a suggestion for a specific.
We're actually like working on a review paper or a methods paper / review, and we're looking at like 20 different methods. So I'll have more to tell you in a month or two.
That'd be great.
And they actually, majority of them run very quickly. So you could actually do a lot of them if you wanted to. Just actually having the LD score regression cross-trait heritability is actually helpful in terms of guiding what phenotypes you might want to incorporate into in terms of a pleiotropy analysis.
We'd love to hear that. That would be great to share. And that goes, by the way, anybody else on the call, we are a big push is to implement new methods. We are very, very responsive to suggestions people have, probably overly so. So we'd love to get ideas like that. That'd be great. Any other comments or questions on anything we've talked about today? And even simple questions like what is LD score regression are fine too. We get that there's a wide range of backgrounds and interests here. So we're happy to elaborate on anything that we've discussed here. And as Mackenzie said, you can email her as well. All right, going once. Anything last things we should tissue enrichment of SNP seem to be too broad rather than cell type specific. Yes. So good point. So this alludes to the fact that right now, all our data sets are grouped into higher level tissue types, into these, I think it's 24 different tissues. That's a criticism that most of the people we showed this to have said that, like, I don't want to see enrichments for pancreas, I need to see islets. So fortunately, all the data sets are available with lower level annotations. And as Patrick alluded to, the next step is to extend from only running over these 170 higher level tissue types to running across the 2000 lower level. So once those are in, which should hopefully be by our next release, I don't know if we'll make it, but end of June we're targeting, you will be able to then see like very cell type specific. And then we'll also be able to leverage the single cell data sets that have been grouped into cell types as well. And we know that's a big limitation. So one of the benefits of LD score regression is it even allows us to do that. Gregor was too slow for us to even consider that. So what we focused on thus far is replacing the old functionality with Gregor with LD score. Now we're going to speed it up and we'll be able to get very, very granular enrichments. And then it'll just be a boatload of data to look at. All right, yeah, chat is fine too if you're shy and you don't want to say it. All right, Mackenzie, any announcements before we go then, like about the next focus group or webinar, or do we just send that out over email?
So our next focus group that we have will be in 2 months, but we do have the Knowledge Portal webinar from 12 to 1 on June 8th, and we'll send information about all of that out through our connections. So if anyone has any questions or they would like the Zoom link, please feel free to reach out to me. I can relay any of the messages to the teammates, or if you just have a question for me in general, please feel free to do that.
All right. Thank you everyone for attending, and thank you, Patrick, for all the work. It's a human's amount of work. And so please, and again, contact us if you're interested in helping explore any of the data, whether that's broadly across the method, specific to a trait, specific to a cell type. We are active. I think there's a lot of publications that could come of this. And so we'd love to have your help. And also if you have a method idea, please be in touch. Thank you all, and we'll see you soon.
Thank you.
Take care.